our first pitch, and it's Lorenzo from Triton Bio.
Hey guys. All right, let me get my slides up on here. Okay, just a quick check. Can everybody see the title slide?
Yes, it's great.
All right, so good afternoon, everyone. My name is Lorenzo D'Amico. I'm the founder and CEO of Triton Bio, and we're working on sort of a new generation or a new class of infectious diseases diagnostics that we're calling microbiome diagnostics. So perhaps a timely, didn't mean to laugh, sort of a nervous laugh. Everybody, I think, on the call is keenly aware of what's going on in the world, and we've been actually working on this problem for a while now. So infections that are either triggered by a virus or involve bacteria or fungi can become incredibly deadly. And so worldwide, the mortality rates are pretty staggering. One of the big problems that plagues the medical care of patients who are afflicted by an infection is identifying the appropriate antimicrobial therapy. In the case of bacterial infections, which could be caused as a secondary infection from a virus, is that every hour that goes by, the chances of mortality increase dramatically. And needless to say, the consequences of this economically are also staggering. And so actually, severe infections can be the most expensive hospitalization category, at least in the United States, that's true. And so really at the core of the problem is actually sort of a technology problem. So the clinical presentations of an infection, whether it's a bacteria or a fungus or virus or a mixture of these organisms, are roughly the same, right? Fever, chills, elevated temperature, there are many others. And so the most definitive way to make decisions about antimicrobial therapy, that's deciding which drug to give the patient to combat that infectious agent, is to do a laboratory workflow. So what I've shown here are the different steps that are involved in that process. But the main point of this slide is to show that it's necessary to first confirm which organism type is causing the infection, and then to identify, and then finally you have to determine sort of physiologically in vitro which drug most effectively kills that organism. And that workflow currently takes many days. And so that number should raise an alarm in your head, because I just mentioned that every hour that goes by, the chances of dying increase by almost 10%. So taken together, what has to happen then today is what's called empiric therapy. That's really what that means, is that doctors are sort of doing a guess and check while they're waiting for lab tests to come back. So our solution to this problem is to try to speed up that laboratory workflow as much as possible. So keeping in mind with the patient first, understanding that they face extreme risk with any laboratory delays, we're developing an instrument that would be sort of like a benchtop instrument that could be deployed either near the patient or in an ER setting laboratory, or as close as possible, and can rapidly provide guidance to the clinical management of patients with infections. I don't want to spend too much time on this technology slide. Suffice it is to say that the assay that we're developing has to accomplish three things in order to supplant these slower culture methods. It has to enumerate the pathogen levels, has to provide some identification, whether it's at the class level or even at the species level. But most importantly, it has to report the physiological response of organisms that have been isolated from the patient samples as they were responding to antibiotics in an in vitro format. And so to do that, we're developing this sort of two-shot assay, which broadly we call an electrokinetic chromatography technique. And so the machine would auto-sample directly from the liquid sample that's obtained from the patient, whether it's blood or urine or a bronchial aspirate. And in these two different tests, sort of in sequence, it would determine the identity by measuring the intrinsic physical properties of bacteria or fungi or viruses, by looking at their electrokinetic behavior in our device. That same parameter of the organism can be used to evaluate its response to a drug in vitro. So we call that a cytoelectric shift assay. And so together, these two techniques, we believe, can be used to accelerate the laboratory workflow. So I'm leading the team right now, but I'm supported by a great group of people who have a lot of experience in the different aspects of what we're doing in our business. So from engineering expertise, Peter Gascoigne brings many decades of working in this industry. Nadim has extensive experience in virology, microbiology, and then Jill was actually pretty high up in business development activities for a diagnostics company called OraSure. And the three of them have been really helpful in getting this going with me. So moving over to our business model, the way we see that the technology can be commercialized is there's a couple of different revenue streams that is mostly driven by the recurring revenue is consumables. So these disposable cartridges that would hold the sample as well as the reagents needed to run the assay. A typical lab, we interviewed over 300 key stakeholders in the microbiology testing industry. And through those sort of customer interviews, we've come up with some estimates on the average lab testing volumes. And so typically we imagine that a lab will run about 5,000 of these tests per year. To run our test, they would need to acquire an instrument. Most labs end up buying two. They have to have backups. And then they also, almost every single company that we know is playing in the space sells maintenance contracts. And what's unique about our technology is similar to an existing technology called MALDI-TOF. So we believe we can also make money by selling the license to software, which provides the interpretation of the measurement so that it's human readable, easy to interpret for clinicians and laboratory personnel. Now, the pricing model, we've deduced that by working backwards from the CMS lab fee schedule. So I mentioned that the instrument is being designed to run 3 specific measurements: count, identity, and antimicrobial susceptibility in an automated fashion. What that means for our customers is something called code stacking. And so by taking these reimbursement codes and adding them, we can work backwards, making certain assumptions about the projected transfer prices through distributors to work out our price, which we've shown here on the slide. And yeah, so thinking about our next steps. So right now we have funding from the National Science Foundation. That's really to do some of the engineering work and some preliminary testing with different lab isolates of microbes, focusing mostly on bacteria right now. And then we also have a contract with the Baylor College of Medicine to use our prototype system to evaluate Cryptosporidium parvum that is infecting patients who are immunocompromised. It's a partnership actually with the MD Anderson Cancer Center in Houston. And so there, the problem really is that Cryptosporidium is actually very difficult to culture. And so they're looking for an alternative strategy to conventional culture methods. So it's a kind of an ideal partnership for us right now to try to de-risk the technology and help out some folks who are doing some highly relevant clinical research. Thinking about our next steps and what we really need to do, I've actively been trying to build a team around this technology and this business. We are housed at the Lundquist Institute in Torrance, California, just upstairs from the infectious diseases department. And so we're trying to build some bridges there to gain access to samples and microbial isolates. And then of course, trying to bolster our intellectual property portfolio. And with that, I may have gone over time, I apologize, but I'm happy to take any questions. Thanks.
All right, thank you, Lorenzo. If anyone's got any questions, please go to the Q&A button down at the bottom of your screen, and I will moderate some of the questions for Lorenzo, and he can answer them verbally to you while you listen. While we're waiting, Lorenzo, I will ask a question. Sure. How relevant, given the COVID-19, how relevant is this for determining, say, a viral load in a patient or something like that? Can we determine whether they've got a regular type of influenza or they've got SARS-CoV-2. Can you let us know a little bit more about that?
Yeah, I'm thinking a lot about that lately. Thanks for that question, Jim. So I see the technology being relevant in two ways. What we know is that a lot of patients are actually succumbing to the secondary infection. And so it's that age-old problem trying to figure out which antibiotics can help prevent the systemic spread of bacteria that are already in the lungs. And so the virus we know is damaging the lung epithelium, enabling an infection to spread from the lung, bacterial and viral. And so if one could help mitigate the bacterial infection, perhaps patients could be stabilized before they suffer severe sepsis, multi-organ failure. So the second way that I've thought about this technology being relevant is actually in the context of research. So we know that these electrical properties we measure now, they're not unique to microbes. We could actually support folks who are screening antiviral drugs using cell line models. And so the technology would be employed to look at how protected a mammalian cell line would be to viral infection in an in vitro assay. There's a third concept that we've actually been talking to people at Baylor about trying this out. In terms of timing of the impact, it would probably be too late, but being able to look at how neutralizing antibodies are changing the electrical properties of viruses themselves is definitely intriguing, especially if our assay, these electrical properties change in minutes, not hours or days. And so there's an opportunity there perhaps.
Have another question for you, Lorenzo. How much funding have you received and what are you looking for in regards to investments at this time, if at all?
Yeah, right. So right now we have a total of $800 and some odd $1,000 non-dilutive funding. The $600,000 from Baylor is actually spread out over a few years. We are trying to raise money, at least $500,000, mostly to bring on some more personnel, specifically sort of a lead scientist to help with the assay development, somebody who has experience in antimicrobial susceptibility testing already, and also a couple of technicians to help with data generation. So yeah, that's sort of where we're at right now.
Okay. I have another question for you while we're waiting for any other ones to come through. What about non-pathogens? What about things like analyzing cell lines for cell-grown cultured meat or alternatives in agriculture, things like that? Do you have any ideas around those?
Yeah, actually, I love that idea. I
always ask the relevant questions. You know that.
No, I appreciate it. Definitely. I mean, the electrical properties of cells have largely been unexplored, and I think that as things like cellular agriculture, alternative meat products, like you mentioned, microalgae, these are highly relevant to multiple industries. So new ways to analyze cells, new ways to understand their physiology and how it changes in real time. I think they're extremely valuable in even things like breweries. But what sort of where I've come from in my own training and the experience I've had with this technology, we've spent a lot of time thinking hard about the clinical problems, but the microbiome at large is a very exciting place to be right now. In a way, I think COVID kind of highlights that. There's a lot we don't understand still about microorganisms.
I have another question here. Can this testing be done outside of the hospital referral? Wondering if there's a timely way of testing public. I personally know that there are issues with personalized testing and the efficacy of that, and I know that there's guidance from the FDA on taking tests at home and stuff. But what are the other options that you have outside of the hospital environment?
Yeah, historically, okay. So the short answer to that question is that we've largely focused on sort of hospital-based and centralized testing. The reason for that is that folks who are suffering from severe infections will be hospitalized, and that's really the setting of care. And so I think that that's the most appropriate setting for testing. That being said, The ER laboratory setting is historically is a place where less complex tests would occur. And so I think there would be value in being able to deploy maybe a second generation of this technology in an ER setting. But again, I think there it's really a question of triaging. And so looking at other markers of the disease, inflammatory response, perhaps the DNA of microbes, that could be done in minutes, and it could be sort of a precursor to this assay.
I think a last question is what we've got time for. Biggest obstacles that you face currently. Is there anything that is a barrier that's holding you back right
now? Maybe other entrepreneurs can relate to. I don't know. It feels like there's a lot of barriers. I'd say right now, I think that we are still a bit resource constrained, even though we have some funding. I think that having more funding, not a ton of money, but having a bit more money will be good for me to help build this team out a bit more. So the other folks I mentioned, they're mostly part-time. I think that having a few people who are fully committed to the technology development effort would be helpful even for me as the CEO to free me up so I can start spending less time in the lab, more time forming partnerships with clinical partners, right?
Yeah. Great. All right. Thanks, Lorenzo. Thanks very much.
Thanks, everybody.